Hello everyone, welcome to sqadia.com. Our topic for today is lab diagnosis of viruses. We are at section 1 of this topic, and in this section we'll be discussing the introduction to diagnostic methods. So we'll start by just a brief overview of diagnostic methods which are commonly employed in order to detect viruses, and then we'll move on to the common approaches of diagnosis, and then we'll discuss the identification in cell culture in detail. So there are different methods through which we can diagnose a particular viral infection inside a cell. The first and probably the most important of those diagnosis method is the direct examination of a specimen. We know that understanding of anything or any new thing especially begins from just observing it. And so a direct examination is can be one of the most crucial phase in order to detect a type of infection inside a cell. In case of viruses, this examination is not done to actually visualize the presence of virus inside the cell, but it's most commonly employed to see the damages that are caused by the presence of a virus inside your cell. Then there's direct detection, indirect detection. In this case, we usually see different effects of viruses that may be caused inside a cell, or different altered functions that are present which can help us identify a type of virus which may be present inside a cell. In this, we can also see the different components of viruses, like different viral antigens or different viral nucleic acids, which might be present inside the cell. We can also see production of certain proteins or presence of certain proteins and nucleic acids, which are only present in those particular cells where the virus is present and are absent in other cells. So we can indirectly detect virus by detecting a number of various factors which shows the presence of virus inside a cell. And then there are certain serological methods that can be employed, and then that can help us to identify and detect a viral infection. Okay, so some of the common approaches to diagnosis are basically the first one, which is used, is the identification of virus in cell culture. We know that viruses do not grow outside of the body, and there are a particular type of cells that are required in order for a virus to multiply inside the cell. And if the virus is actually active and multiplying inside the cell, then and only then can we actually identify the presence of virus in a cell. So the first one is the detection of virus in cell culture. There are certain difficulties in this particular diagnosis approach. We'll be discussing that later in this lecture. Then there is microscopic identification, which is done directly on the specimen. The specimen can be the blood of the patient. The specimen can be any body fluid of the patient in which there might be a chance of presence of viruses. Then there are serologic procedures, which basically detect the rise in antibodies which are present in your body. And this rise in antibody titer can actually explain or can actually help us identify the type of viral infection that might be present inside your body. Then there is presence of IgM antibody. This is a specific antibody which will be present because of a specific type of viral infection. We'll be discussing that later in this lecture. Then there's detection of viral antigens in blood or body fluid. Again, there are certain viral antigens which are specific to the type of virus, and we can develop antibodies against those antigens and can detect them through different serologic procedures. And then there is detection of viral nucleic acid in the patient's blood or the patient's cell. As I explained earlier, these are the type of nucleic acids that differentiate or that have a different effect on the patient's cell and are different from the patient's nucleic acid. So uh, we can identify them on the basis of, for example, double-stranded RNA, which might be present in the patient's blood, etc. So all of these are the common approaches, and there are certain approaches that are more specific to the type of virus. There are certain approaches that actually help us identify the stage in which the viral infection is. And so there are different specificity levels of each of these diagnosis approaches. As explained here, you can see that there are certain specific approaches which are direct methods, which include the virus isolation. This can be done in cell culture. Then we have the virus nucleic acid detection and the viral antigen detection. And then there are indirect methods, which are usually serological methods that help us identify the presence of IgM and IgG antibodies inside the patient's blood or body fluids. And we can identify these antibodies there and thus identify the type of infection which might be present. So the first one is the identification in cell culture. We know that for a virus to be able to grow and replicate, it requires a cell. And thus, cell culture is important because outside of the body, the virus will not be able to replicate, and we cannot identify whether or not a virus is present inside a patient or not. So, for that, development of cell culture is important. There are many viruses which are inactivated at room temperature. In certain instances, you can keep the sample in which the virus might be present at 4 degrees centigrade, but not for a really long time. And so, in case of such samples, it is important that you use those samples or you grow the cell on the cell culture plates immediately so that the virus may not get inactivated. Um, Once the virus starts growing inside the cell culture, the virus growth is seen because of certain changes that are caused by the virus inside the cell. These changes are called as cytopathic effects, and this is basically the change in the appearance of the virus infected cell, which will help us identify and differentiate those cells from the non infected cells. This change can be in the form of size, shape, or fusion of the cells to form multinucleated giant cells. This is also known as syncytia. And this is basically the identification of the virus and the presence of virus inside a cell and how that virus causes changes inside a cell. Um, and those changes can help us identify the type of virus and can help us identify the infected cell from the non-infected cell. So this is basically a manifestation of the viral infected cells, and these cells are usually dying or are already dead because of the viral infection. And those manifestations can be detected in the cell culture, and we can identify the growth of the cell. The problem with this particular technique is that it's really hard for certain cells to be grown in a culture, And so, if a virus is if a virus particularly grows in a particular type of cells in your body, then it's important to culture only those type of cells, because the virus may not replicate in different cells, or cells other than they usually infect. So, developing those cell cultures is one of the more difficult or more expensive tasks in such identification techniques. The easiest virus to detect in cell culture or through cell culture technique is actually a bacteriophage, because as we know, phages are the viruses that infect the bacterial cell, and bacterial cells are one of the easiest cell cultures to grow. So it's really easy to identify those bacteriophages inside the culture. But other than that, developing of a culture and then identification of presence of a virus in that culture is no doubt a very direct method to detect a virus, but it is time consuming and it is expensive. There are different types of cytopathic effects, or different types of manifestations that can be seen that can help us identify a virus inside the cell. The first of these types of culture is the heme adsorption. This is basically the attachment of erythrocytes to the surface of virus infected cells. So if there is presence of viral infected cells, the erythrocytes will attach to those particular cells, and we can easily identify those cells, and we can easily visualize the change. This particular technique is limited only to the virus that have a hemagglutination protein in their envelope, because if virus lacks a hemagglutination protein in their envelope, then the erythrocytes won't attach to the cell. And so this particular manifestation, or this particular identification technique, is used only for those viruses that have hemagglutination protein on their envelopes. Viruses such as belonging to the mumps or the parainfluenza or the influenza family are basically all of the viruses that contain the hemagglutinin protein on their coats, and so we can identify these type presence of these viruses in cell culture through heme adsorption. Then another form of cytopathic effect that can be caused by a virus is the interference, which is the formation of the cytopathic effects by a second virus. The example will help us explain and help us understand this phenomena in a better way. For example, we have a rubella virus, which does not cause a cytopathic effect, but this can be detected by interference with the formation of cytopathic effect by certain enterovirus. This means that if a cell is infected with a rubella virus, then we won't be able to identify it in cell culture, because rubella virus does not cause cytopathic effects in the cell. But if we infect those same cells with any of the enteroviruses, then those enteroviruses will cause interference and will cause cytopathic effects in the cell. And this is like the indirect identification of presence of rubella inside the cells. So this phenomena, or this type of cytopathic effect, is known as interference. Another type of cytopathic effect that can be seen is the decrease in acid production by the cells that are infected, or by the cells that are infected and are dying because of the infection. This can be detected visually by a change of color in the phenol red, which is a pH indicator in the culture medium. and this is mostly used to detect enteroviruses. So if a particular type of cell culture or a particular type of cell is infected with enteroviruses, then we can add a pH indicator, which is in this case a phenol red, in the culture medium. And once this pH indicator is present inside the medium, there will be decrease in acid production by the cell culture that will result in a change in color of that medium, and this will help us identify the type of identify whether or not the cells are infected. And enteroviruses are almost all the enteroviruses are detected by this method. This figure here shows the phenomena of hemagglutination, which is basically when the virus infected cells accumulates or attach erythrocytes to them, and you can see the phenomena of agglutination. And this agglutination will show that yes, there are viruses that are present inside the cell, and those viruses are causing the erythrocytes to attach to the cell. And so it'll be easily detected or visualized. So in this section, we covered the overview of diagnostic methods. We saw the common approaches to diagnosis of viruses, and we finally explained how we can identify different types of viruses in cell culture. We can use different methods like heme adsorption, like interference, and there are different ways in which virus can even change the color of the medium with the help of pH indicators, and thus we can visualize that change in the cell culture, especially in the cells that are infected by viruses, and thus we can differentiate those cells from the normal cells. That's all from this section. Thank you for watching sqadia.com.